Okay, so good afternoon, everybody. Today my presentation is on antivirals. So basically, antivirals is classified into two types. That is antiretrovirals and antiretroviral agent and non-retroviral agent. So under non-retroviral agents comes anti-herpes and anti-influenza. So I'll start with antiretrovirals first. So can you tell me some examples or any classification about antiretrovirals? Okay. Okay. So basically, we have five class for antiretrovirals. The first class is reverse transcriptase inhibitors. Okay. So that is divided into NRTIs and NNRTIs. That is nucleoside reverse transcriptase inhibitors and non-nucleoside reverse transcriptase inhibitors. That is the first class, reverse transcriptase inhibitors. Second is protease inhibitors. Third is integrase inhibitors, and fourth is fusion inhibitors. Okay. So first I'll explain the life cycle of HIV so that it's easier. So this is the cell membrane, the human cell membrane, and we have a CD4 cell. Okay, this is a CD4 cell. So we have a HIV virus, whatever. Okay, with the RNA and all. So this part, okay, this is a part where this part is called as GP41. Okay, and this part is called as GP121. Okay, so we have now this GP41 is the one which fuses with our CD4 cells and it gains entry via the cell membrane. Okay, and in this cell membrane we have two more structure called as one structure is called as CCR5 and CXCR4. Okay, these are the two structures which is present in the cell membrane. So this GP41 have to fuse with this, either of this, and then only it can gain entry into the cell membrane of the human cell membrane. Okay, so then this now after fusion, this HIV virus is entered into the cell membrane. So what is it carrying? It is carrying the RNA. Within that, it is having the reverse transcriptase enzyme. The HIV virus only is having reverse transcriptase enzyme and this RNA. Okay, and it's got So now once this enters, the RNA is released. That once this has been once this fusion has happened, this RNA is being released into the cell membrane. Okay. Then and at the same time it releases RNA, and at the same time it releases reverse transcriptase. Okay, so this RNA in the presence of reverse transcriptase enzyme gets converted to DNA. Okay, this is a viral DNA, not the human DNA. Okay, this is a viral DNA. Then this DNA enters into the nucleus. This is a human nucleus. Okay, this enters into the human nucleus DNA. Which DNA is this? This is the viral DNA. Okay, and inside the nucleus what do we have? We have human DNA, right? So we have our human DNA. So what happened? This viral DNA, this viral DNA is a single stranded DNA. Okay, so this will fuse with the human DNA. Okay, I'll represent that with a different color. Okay, so this is the viral DNA. Viral DNA. It will fuse in the presence of an enzyme called as protease enzyme. Integrase enzyme. Integrating, right? It is getting inside. So it is integrase enzymes. Okay. So now this is the DNA. This is the DNA which contains both the viral DNA and the human DNA. So now this viral DNA, you know what is transcription, translation? What is transcription?
DNA to RNA.
Okay, transcription is conversion from DNA to RNA, and translation is from RNA to proteins. So inside this now transcription happens, and this gets converted into mRNA. Okay, it is a viral mRNA because now the viral DNA is attached, right? So this is called as viral mRNA. Okay, and then this gets released into the cytoplasm, and in the cytoplasm we have ribosomes. Okay, we have ribosomes, and this viral mRNA undergoes all these processes within the ribosomes. It undergoes all this maturation process. This process is called as translation. Okay, conversion, and this mRNA gets converted into proteins. Okay, and these proteins are both. There are two types of proteins. That is structural proteins and functional proteins. Okay, this functional proteins and structural proteins are being produced, and this gets incorporated, and these proteins get matured. Okay, and these proteins get matured in the presence of an enzyme called as protease enzyme. Okay, the structure and functional proteins, and there is new HIV virus which has been formed. Okay, and this later on enters into the Golgi apparatus. See, this part you should all learn. Okay, this part, this viral DNA gets converted into just forget about this integrase, but then in general most of the bacteria or whatever is entering, this viral DNA will get converted into viral mRNA, that is transcription, then it will undergo translation to proteins, and then it will undergo maturations, and it gives rise to new virus. Okay, new virus, and this fuses with your and this enters into your Golgi apparatus. Okay, within the Golgi apparatus it undergoes packaging. Okay, once it undergoes packaging, this see, now all what you have is proteins. Once it undergoes packaging, you'll have a daughter cell with the proteins and all that. Okay, and this fuses with your cell membrane, and it releases the virus outside the cell. Okay, so this is what is happening. This is a basic cycle. Okay, most of the viral, so first step is viral DNA will get converted to mRNA, that is the mRNA, then it will convert it to protein, then via the ribosomes, then this protein enters into the Golgi apparatus, gets matured, and then it fuses with the cell membrane and releases new viruses. Okay, so that is like the common mechanism for all the virus. And in case of HIV, you have integrase which helps to bind into the human DNA. Then we have protease which helps in the maturation of the proteins of the structure and the functional proteins. And here, okay, so now coming to the drugs part. So first we have one class of drugs called as fusion inhibitors. Okay, so that is the one which inhibits the cytochrome CCR5 and cytochrome CXCR4. Okay, the first class is fusion inhibitors. The examples, one drug is known to inhibit cytochrome CXCR4. Okay, that is enfuvirtid. Okay, which inhibits CXCR4, and the drug which inhibits CCR5 is maraviroc. Okay, these are fusion inhibitors, the first class. Okay, so the first drug is fusion inhibitors. Next come the second drugs which inhibit this integrase enzyme. Okay, that is IIs, integrase inhibitors. The examples of integrase inhibitors are dolutegravir, DOLUTEGRA. So gravir, dolutegravir. Then we have raltegravir. Then we have bitegravir. Okay, so integrase inhibitor, the synonym is gravirs. Okay, all the gravirs. Next set of inhibitors is this enzyme which prevents the maturation. So that is the protease inhibitors, that is the PIs. Okay, so the examples of this is atazanavir, ritonavir, all this. So the synonyms is navirs. Okay, atazanavir, indinavir, lopinavir, rotinavir, nelfinavir, saquinavir. So all these navirs are protease inhibitors. So first is fusion inhibitors, second is integrase inhibitors, third is protease inhibitors, and we have one of the main thing, that is the reverse transcriptase inhibitors. Okay, so that is RTIs, reverse transcriptase. Again, we have two types, NRTIs and non-NRTIs. So what is the difference between nucleoside reverse transcriptase and non-nucleoside reverse transcriptase? Nucleoside reverse transcriptase, it acts as competitive inhibitors. That is, it will compete with this, it will act as a nucleotide. So it will compete with this and thereby prevent the formation of the DNA. Whereas non-nucleoside NNRTIs, it will directly go and inhibit this enzyme. It won't go to compete. Okay, that is the difference. That is NNRTIs. So coming to NRTIs, the example is zidovudin, abacavir, lamivudin, stavudin, tenofovir, and diadenosine. Okay, so here we don't have any synonyms because it goes ups and down. Whereas in NNRTIs, it is nevirapine, efavirenz, itravirin, and delfavirdin. So we have this virdin, vir. Okay, so that is, so the four classes of antiretrovirals is, first is fusion inhibitors, reverse transcriptase inhibitors, integrase inhibitors, and protease inhibitors. Okay, so that is about antiretroviral. So now I'll go with anti-herpes virus. Okay. Okay, I'll go with anti-influenza virus. Okay, so we have a cell membrane. This is a human cell membrane, okay, and we have a virus. This is the influenza virus, okay. Okay, so this okay, just imagine all this. So this influenza virus is having three main parts. So first part, this is called as neuraminidase. Neuraminidase, okay, that is one enzyme. Next enzyme is hemagglutinin. I mean, not enzyme, a structure, okay. Hemagglutinin, this is a protein, hemagglutinin protein, and there's one more thing called as protein ion channel, that is the M2 channel, okay. So these are having different functions. I'll let you know. So on the cell membrane, there is something called as salic acid, okay. There is something called a salic acid. So this H2 enzyme, this protein is there, right? So this fuses with this and gains entry into this, into the cell membrane, into the human cell membrane, and there is uncoating. That is, this is a virus, right? So this is the influenza virus. So this fuses with this chain and enters, and it releases the RNA. Okay, so this process is called as uncoating. So after that, this enters. So now this is a viral DNA. I mean viral RNA. Okay, so this as usual enters into the nucleus of the human cell. Then in human cell, this viral RNA gets converted to and inside this nucleus we have viral mRNA. Okay, I mean human mRNA we have. The human cell, that is the human mRNA we have. And this human mRNA, you know, all this 5' thing and all. So what happens, this viral RNA have to become like that. It should undergo that post-translational process, all that 5' thing and all. So this will happen in the presence of an enzyme called as endonuclease. Okay, endonuclease. And this viral RNA gets converted to 5' RNA. In the presence of endonuclease. Then same process, that is this now this is a viral mRNA, right? So this viral mRNA, that is a 5' viral mRNA, will get into ribosomes, undergo all this translation, and proteins will be formed. Then proteins will get converted to then proteins will get enter into the Golgi apparatus, undergo packaging, and will be released as vesicles. Then that vesicle will fuse with the cell membrane and release the virus. So the same function is going on. Okay. So now after coming here, now at the end, what I'm telling you, it went into the Golgi apparatus and we have packed vesicles. Okay, this is the viral DNA. So what this have to do? This have to again gain exit, right? So here again we have the salic acid. Okay. So what happened? This will come out with along with the salic acid, but it can't uncoat. I mean, it can't release the new immature virus which is present inside this package. So what happened? We have this neuraminidase, right? So this neuraminidase cuts off this bond with the salic acid, and that is how it will help in the release of new virus into the cell. Okay, so now this is the process. So what is the main structure? First is uncoating, next is conversion, that is conversion of formation of post-translated mRNA, and next is exit. So we have drugs which prevents these steps. So first is drugs which prevents the uncoating, that is only if the virus, that is the RNA, is gaining entry into the cell, all this process happens, right? So the first drug which prevents this uncoating is amantadine and rimantadine. Okay, amantadine and rimantadine. So they are called as uncoating inhibitors. Okay, the first class is called as uncoating inhibitors, that is amantadine and rimantadine. Uncoating inhibitors. Next we need something to inhibit this endonuclease. So that is called as endonuclease inhibitors, that is balexavir. We don't use this much, okay, balexavir. Okay, here it is amantadine and rimantadine. Okay, so first is amantadine, that is uncoating inhibitors. Next is endonuclease inhibitors, and third step is we have this neuraminidase, right? Because of the neuraminidase only, it's undergoing release of the new virus into the body. So we have something called as neuraminidase inhibitor. That is our one and only favorite, oseltamivir, zenemavir. Okay, so the best part about all this is all these drugs should be used within 48 hours post exposure. All these drugs, any drugs. Okay, but then the most common what we use in this is neuraminidase inhibitor, which is oseltamivir, zenemavir. Okay, again oseltamivir, which is common. So this is a classification of anti-influenza agents. So can we come again? So antiretrovirals, first we have fusion inhibitors. RTIs, reverse transcriptase inhibitors. Then we have integrase inhibitors. Then we have protease inhibitors. So we have 4 classes of drugs under antiretrovirals. Next is under anti-influenza, we have 3 classes. First is uncoating inhibitors. Next is endonuclease inhibitor. And finally we have neuraminidase inhibitor.